Welcome to the lecture on DNA and mitosis. We're not going to spend a ton of time on DNA. We'll talk real briefly about the structure and a little bit about how it's duplicated, and then we'll spend most of the time looking at cell division or mitosis, which is a type of cell division. So you can see here on the title page, we have a couple cute little beautifully drawn images for you. We've got two cells separating over here on this side, and a nice DNA molecule on this side. That's my beautiful artwork for you for this chapter. Apologize for the cough. So moving on here, let's look at the structure of DNA here. So we're looking at a DNA molecule here. Now some key characteristics of DNA is you see the double helix. Right, two strands, so a double-stranded helix. Helix referring to the spiral formation of the DNA molecule. Now, what makes up each strand of DNA? Well, DNA is a nucleic acid, right, which is in its name, deoxyribonucleic acid. And what makes up a nucleic acid, if you remember back to chapter 3, is a nucleotide. Nucleotides are made up of, and I've drawn one over here for you. So I've blown this up here. So I've blown up this segment of DNA over here. And nucleotides are made up of a phosphate and a sugar. Now DNA has its own type of sugar, which is part of its name, deoxyribose. That's why I have D-Shug here. D-Shug. Sounds like a rapper. So D-Shug makes up the sugar portion of the nucleotide that makes up DNA. And then again you have your phosphate up here, okay, and sugar down here. And then coming off, which would be actually in the middle, so they look like this in the middle, and they actually help hold the DNA together. It's what helps create the DNA molecule is the nitrogenous base, and that's or nitrogen containing base, and that's why I have N base. So it's a nitrogen containing base. And there's four types that you'll see in DNA. That's adenine, guanine, cytosine, and thymine. Okay, and you can look those up in the book. Okay, and don't worry too much about spelling, just you need to know them. Okay, and what happens is they pair up accordingly, and they have hydrogen bonds between them here. And that's what keeps the DNA bound together. So you have these hydrogen bonds, which is what those dots are supposed to represent. Okay? And so they're bound together, and that keeps the double helix. Alright, now I'm going to clear this page. I'm going to show it to you again here. So let's say we have a small segment of DNA here, and it's the base A, G, C, T, and now these are connected to their backbones, right? Then each nucleotide backbone. So you're going to have a phosphate and a D-Shug, right? A phosphate and a D-Shug, and so on and so on. Okay? And so that's your backbone of the DNA. Okay? And then your bases out here, if you look at DNA like a ladder, they're like the actual rungs you grab onto. So the backbone's like the side rails, and the rungs you actually grab onto as you go up the ladder is your nitrogen bases. If I have A, G, C, and T on this side, the other side of my DNA that it's going to be bound to through a hydrogen bond is specific to those bases. So for example, adenine A binds with thymine T. Okay, and I'll actually use blue still. A and T bind together. Their unique structure allows them to hydrogen bond together. T won't bond with, that won't happen right there, and that won't happen right there, and that won't happen right there. Okay? So thymine won't bond with cytosine or guanine or itself. Okay? Guanine bonds with cytosine. Cytosine bonds with guanine. That's a terrible looking G. And thymine bonds with adenine. Okay? And these are hydrogen bonds. Okay, those are hydrogen bonds. So that's what keeps the double helix together. Now, before we get into cell division, DNA actually has to duplicate. And what happens is you have your, actually I'll use blue. You have your original DNA molecule there, and I know it's not the most beautiful double helix spiral there, but it'll work. It actually has to duplicate. And so what happens is, part of it, at some point, it'll start to peel apart. And so it'll actually look, and let's do this. Let's start over here. So it'll actually then start to look more like this. Oh, and it starts to peel apart. And what happens is we start to copy it, and it'll slowly peel back, and we'll copy the strands. And how we do that is, for example, let's say that right there, this is the segment AGCT. Well, this segment coming in right here is going to be the base pair of that. The complementary pair is what they call it. So this would be, the new strand would be T, C, G, A. It would be the pairs that go with it. So it's not an exact copy. Its copy is actually going to be, the copy of the original would actually be over there. If that makes sense. Okay, so we're just base pairing in order to make two whole new molecules of DNA. And so this would happen all the way back to the DNA molecule. And you would end up, at the end, with a molecule that looked like this. You would have one of the originals with a new one, and one of the originals with the other new one. And that's what happens at the end of DNA replication or DNA duplication. And that'll happen before every cell division. Alright, let's look at before we get into cell division, let's look at the cell cycle. So cells have life cycles. We have triggers in our bodies that actually tell them when to proceed through certain steps of the life cycle. And that's our cell control. And if we don't, that's how we can get into cancer and those kind of illnesses, when we aren't controlling the growth of our cells, or they're not responding to our control. So let's look here at the cell cycle. The segment in green is what we call interphase. Interphase, all one word, I'm just breaking it up here. Interphase. Interphase is broken down into three parts: G1, S, and G2. Okay, G1 stands for gap 1 phase, S stands for synthesis, and G2 stands for gap 2 phase. In the gap 1 phase, it's just a cell's normal life. It's going about its normal functions, it's growing, and it's just kind of think of it like a child. Not really thinking about anything, but just what's happening at that moment. And then, when a child gets into their teenage years and high school years and they start to think about dating and what to do for the future, what college they're going to do they start to prepare for leaving the house. And that's what the synthesis phase is all about. Their preparation. Well, how do they prepare? Well, they prepare by going through DNA duplication. Okay? So they'll actually go through DNA duplication or replication in the synthesis phase. Okay, so you'll get two sets of DNA, two copies of DNA during this phase of the cell cycle. And then after that, it goes back into the gap 2 phase, which is continued preparation for cell division. Make sure the cell has enough organelles and nutrients and those kind of things ready for cell division. It also continues to grow if necessary. Now, a cell spends most of its lifetime in the cell cycle. It's about 90%. Okay, so the majority of its life is in interphase. Now, the last 10% is spent in the mitotic phase. Mitotic / cytokinesis, because they happen together. Okay, so that other 10% is mitotic and cytokinesis phase. Alright, this is where cell division will actually occur. This is where we'll go from one cell to two cells. Alright, so that's the cell cycle. Now we're going to look more specifically at mitosis, or the mitotic phase. So here's a nice image of mitosis. So I'm going to draw or write up a little helpful hint here for you to remember. If you can remember PMAT, that'll help you remember the phases of mitosis. Okay, each one of these stands for the first letter of the phase. So the first one is prophase. Second one is metaphase. I'm just going to put the first part because everybody can remember phase. Anaphase and telophase. Okay, so if you remember PMAT, you'll be able to remember the phases of mitosis. Now, here, these images right here give you a nice look into what's actually occurring in the cell during mitosis. If we look in prophase, what happens in prophase is normally there's a nice little nucleus in here, and it's actually breaking down. So the nucleus that surrounds the chromosomes are actually breaking down. Okay, and you see this arrow here that's pointing to those. This arrow there, the line, is pointing to a pair of chromosomes there. Those chromosomes are two identical pairs, right? It's two identical chromosomes bound together. And what we call those is sister chromatids. Each one's a chromatid, and together they are sister chromatids just because they're the same one. There's just one's a copy, one's the original. And during prophase, the chromosomes will line up in that manner. They'll all join as sister chromatids as the nucleus breaks down. We also will get microtubules or spindle fibers, which are these long yellow lines there. Let me do this. So those yellow lines there right below that red line, those are spindle fibers, which will come into play here in a minute. And they come out of the centrioles, which are these little organelles on the end, which you probably already read about in chapter 4. Okay, so that all occurs in prophase. Prophase is just a very quick preparation for the cell to divide. As we get into metaphase, and we should be fine here. As we get into metaphase, the sister chromatids all line up on the metaphasal plate or the equator of the cell. So if we were to make an equator here, they all line up in the middle, right on the equator. As they do that, the spindle fibers also come out and attach to the sister chromatids. They attach to the sister chromatids at their centromere, and then they'll actually get pulled apart where they're connected. Okay, so where those sister chromatids are connected, they'll actually get pulled apart, but not till anaphase. In metaphase, they just line up, and the spindle fibers attach, or the microtubules attach. Okay, and now remember, sister chromatids are two same copies attached together. All right, and you can see those there as well. So let me get rid of these lines. Each one of these pairs is a sister chromatids. Okay, sister chromatids. All right. Now, as we get to anaphase, what happens is the sister chromatids get split apart, and they go to opposite ends of the cell. Okay, opposite poles. All right, they get pulled apart. The spindle fibers pull them back towards opposite poles. Then, as we get to telophase, a new membrane starts to form, pinch in, which we'll talk about in a minute. The chromosomes get pulled to their opposite poles, and we start to get a nucleus forming around the chromosomes. The chromosomes will unwind into their chromatin form. What that is, chromosomes are a nice, beautiful organization. They're condensed DNA. When they're not going through cell division, they're actually more strung out all over the place. They're not condensed, and that's called chromatin. Okay, so when they're not going through cell division, they'll be in the chromatin form. All right, so that's what happens in telophase. The DNA gets pulled to one side, starts to unravel, nucleus starts to reform. Basically, everything opposite of what happened in prophase. Because in prophase, the chromosomes condense, the nucleus breaks down, spindle fibers start to form. So in telophase, we're winding down rather than beginning. Okay. Now, as we get into telophase, cytokinesis also starts to occur. Okay, and cytokinesis is not considered mitosis because mitosis refers to the division of the nucleus and creating two new copies of DNA in different cells. Cytokinesis is the actual division of the cytoplasm, or the final separation of the cell into 2 cells. And so at the end of mitosis there, we saw a picture that looked like this. And what happens in cytokinesis is you'll actually, the microtubules of the cytoskeleton will actually create what we call here a cleavage furrow. And there's a picture of one in the PowerPoint. And what that is that's where they're actually going to pinch apart. So these will actually pinch apart at that point and become their own cell. And that's how it happens in an animal cell. So our cells, this is how they go through mitosis and how they go through cytokinesis and divide. And that happens in telophase and then after telophase. They happen kind of simultaneously there. Now if it was a plant cell, it would happen more like this. Let's say we have the plant cell here. The DNA has moved to 2 sides. You got our DNA in here. What happens is you get these cell plate vesicles in there, and they start to form cell wall in the middle. And then pretty soon it starts to get closer together and closer together and closer together until all of a sudden a cell wall just forms right in the middle. And then you have 2 cells. 1, 2. Okay, 1, 2. And that's the end of cytokinesis. So that's mitosis. Now one thing I want to talk about before we end here is, because it'll become more important when we get into meiosis, is mitosis is the division of 1 cell into 2, okay? And you maintain the same number of chromosomes or copies of DNA from the beginning to the end. And so what we call that is you start with a 2N cell or a diploid cell, and we're ending with exact copies, so that at the end we're still going to have diploid cells. Okay, so make sure we remember that. We start with a diploid cell, we end with 2 diploid cells. Okay, we start with a parent cell, we end with 2 daughter cells that are diploid. When we get into meiosis, we're splitting down into half the number of chromosomes, so it'll get more confusing. So I want to make sure we understand that before we move on to meiosis. Okay, so in summary, mitosis, cell division. You go from 1 parent cell to 2 daughter cells. Parent cell is 2N, daughter cells are 2N. The steps to get there, PMAT. Make sure you remember those. And then cytokinesis has to occur as well in order for you to get full division. Alright? Make sure you also look over the rest of the lectures, meiosis and genetics associated with this exam. Take care.